very good morning and happy Mahashivratri to one and all. Today we'll start with the session 1 of day 3 of GIAN course, and may I now request Dr. Dinesh Handa to please start the session.
Good morning, everyone. Okay, so yesterday we finished talking about metals, ceramics, polymers. They all can be used for medical implants depending on the application. I think next I want to start discussing is slowly some of the issues with medical implants. Okay, so I want to talk about proteins, and then we will talk about blood, we will talk about bacteria in the next 2-3 days. So my goal is to finish proteins today. So let's start with the proteins. Now, when we put any implant in the body, such as a catheter, the first response of the body is proteins come and adhere to the surface of the catheter. And then cells can bind, platelets can bind. If there is bacteria, bacteria can bind to the surface. That can lead to inflammation or infection or clotting or toxicity. But the first, it starts with proteins binding to the surface of the implant. So that's why proteins are very important, and there is a lot of research going on. My lab works on it also. We will discuss, I think, early next week, how to make surfaces that you can prevent proteins from adhering to the surface. Okay, it's a very challenging task because our blood has so much protein, but if we can prevent protein adhesion to the surface, maybe we can prevent clotting of the surface, we can reduce infection of the surface. So the first component is protein, right? Now, not talking about blood material interaction or protein material interaction, but what are some functions of protein in the body, right? So serve structural support role in tissue matrices like collagen, elastin, they all are proteins. Enzymes, these proteins can facilitate biochemical activities in the body, blood coagulation, DNA transcription, antibodies responsible for immune response, hormonal proteins that can coordinate messengers and signals, contractile proteins that are responsible for movement in the body, storage and transport of molecules such as hemoglobin. So proteins have so many different functions, right? So proteins are made from amino acids. They form peptide bonds, polypeptides, and these peptide bonds, just like we discussed polymerization yesterday, amino acids can be considered as monomers, and they are binding to each other and forming peptides and forming proteins. There are these 20 common amino acids, and if you see the structure here, it has an amine group, and it has a side chain, that R group, and it has a carboxylic acid group. And you might have heard some of the proteins, that amino acids that are common, like arginine, lysine, glycine, cysteine, valine, aspartic acid. They are some common proteins, common amino acids, and these amino acids can bind to each other to form different types of proteins. And I'll show you an example. Just one amino acid difference can start causing diseases and problems in the body. So there are like so many proteins, different combination of different amino acids. Everything needs to work so perfectly well, because even if one amino acid gets different amino acids bind instead of one amino acid, now you have a problem, you have a disease. And if you forgot, polymerization for amino acids condensation polymerization takes place to form these peptides, which can lead to these they can form proteins then. Now, proteins can have different structures. There is a primary structure, secondary structure, tertiary structure, and quaternary structure. And you can imagine primary is a simple structure and quaternary is a much more complicated structure, right? So sequence of amino acids in peptide change, polymerizations of amino acids. A simple primary structure example is insulin. It has these 3 disulfide bonds, and this is like a primary simple molecule. Then the next one that's a little bit more complicated is secondary structure, and this is more alpha helix and beta sheets together, and they are forming secondary structure of proteins. The alpha helix, beta strands, and sheets turns are the most prevalent elements of protein secondary structure, which is stabilized by hydrogen bonds between atoms of the peptide backbone. And then we have tertiary structure, which is now making the molecule even more complicated. Proteins fold with polar groups on the outside, and the non-polar groups go inside when this folding happens, so that these polar groups that are outside can interact with the fluid outside, and these non-polar groups are hidden inside, which are more hydrophobic. So hydrophobic interactions, hydrogen bonds, they are common for this tertiary structure of proteins. Ionic interactions can be there also, disulfide bonds can be there. So this is just like leading to a much more, starting to make a more complicated structure in a tertiary structure of proteins. And then we have quaternary structure of proteins, and the proteins are made up of multiple peptide chains called subunits. Now multiple subunits come together to make this quaternary structure, where you have like 4 subunits coming together to make hemoglobin structure here. Okay. So to summarize, these are the 4 different primary structure, quaternary, secondary structure, tertiary structure, quaternary structure. Simple to more complicated has hydrogen bonding and ionic interactions and van der Waals. Simple ones just had those disulfide bonds. Little bit about proteins. Now, proteins have conformation also, okay? And a good example of that is, see, I told you that when there is a surface, there is an implant in the body, protein comes and stick to the surface, and on that platelets are sticking and cells are sticking and bacteria is sticking, right? But that same protein was in your blood flowing, but nothing was sticking to it at that time, right? Because the conformation of that protein was different when it was flowing, and now when it's got stuck to the surface, the conformation changed. So those receptors responsible for binding these things, they get exposed, and now all these things start sticking to that protein. So changes in temperature, pH, ionic strength, binding to surfaces, that can cause protein to get denatured, or certain property of protein that was not there, it came, or the certain property of protein that was there, it got lost. These changes interfere with intermolecular bonds that stabilizes the conformation. Proteins may not be able to revert to its original or native state after this has happened. And an example is, a simple example is like an egg, right? When you are cooking an egg, you cannot just go back now. That example I was talking about of this one amino acid difference, a common example is sickle cell anemia. So glutamate is the normal hemoglobin amino acid that should be there. But if that glutamate is changed to valine, now the person has sickle cell anemia. And in sickle cell anemia, it's a genetic blood disorder where these red blood cells become rigid and they make this sickle shape. And now you can imagine these red blood cells, they are not transporting oxygen properly, and because of their change in the shape, they can get stuck in the blood vessels, which can be really dangerous. And you can also imagine if oxygen is not getting transported properly, the patient can get a lot of fatigue and rapid heart rate, shortness of breath, because they are not getting proper oxygen transfer. Now, other example of proteins, one example is collagen. 25% or more of the total body protein is collagen. There are 28 different types of collagen identified. Makes the structural framework for most body structures, including skin, blood vessels, skeleton. Collagen fibrils can aggregate to form collagen fibers that gives high tensile properties. Now, here are the 5 common types of collagen. Type 1, type 2, type 3, type 4, type 5. These are the common ones, and then there are like 20 or so more collagens that are not so important. But skin, tendon, vasculature, organs, bone, type 1, cartilage is type 2, in liver and tissue, soft tissue, type 3, epithelium, type 4, and cell surfaces, hair, placenta is type 5 collagen. Another important protein, elastin, and like the name elastin, elasticity. So tissues containing elastin behave similar to rubber band to deform and recover without damage. Okay, so it can be stretched and relaxed, and that's giving like elasticity to these tissues. Now we are coming a little bit to the point. Okay, those are good, simple little information about proteins. But now fibrinogen is an important one because we are talking about biomaterials, implants. Fibrinogen is one of abundant proteins in blood. Okay, I think it's 3rd or 4th highest protein present in blood after albumin, IgG, and then maybe it's, I think, fibrinogen. And this is the main protein that's responsible for platelets to get activated or clot on the surface. Okay, so it's a very big protein for people in blood material interaction field. Common form is it's a rod shaped protein, 40 to 50 nanometer in length and width of around 5 to 10 nanometer. It's 340 kilodalton. Many receptor sites to interact with platelets and bacteria, multiple cell attachment sites, and this fibrinogen protein has RGD sequence on it. Now, what is RGD sequence? So, if you see here, RGD is arginine, glycine, and an aspartic acid. Okay, so this RGD sequence or these 3 amino acids form this RGD sequence. So presence of this RGD sequence on fibrinogen helps in cell binding to that RGD sequence on fibrinogen. You know, other protein, for example, albumin does not have RGD sequence, so cells usually don't bind to that protein that easily. Okay, so for our blood material interaction field little bit, albumin is maybe our friend and fibrinogen is more our enemy. Similarly, fibronectin is another protein, and this also has that RGD sequence. So a lot of time, if researchers are working on binding cells to surfaces, they actually coat the surface with fibronectin so they can bind the cells to the surface. It enables cells to adhere, important role in organizing the extracellular matrix. Size is around 440 kilodalton, has affinity for various molecules, contains this RGD sequence that binds to cells. Now, protein surface interaction, adsorption of protein on the surface is the key, right? It's not absorption, it's not getting inside, it's just covering the surface of the implant. Cascade, and bacteria can stick to this protein also. Sure. Okay. Like we said that when you have any implant or a substrate in the body, proteins will come and stick to it, RGD sequence get exposed, cells or whatever, platelets, now they start getting adsorbed on the surface. And when these platelets get stuck to the surface, they start getting activated and they start calling more platelets to come and stick to the surface. And now platelets start interacting with each other, and now the clot formation starts happening on the surface. But it started with the fibrinogen adsorption that started this whole thing. So if we can stop that, we can hopefully stop a lot of things. We will talk, but trust me, people are trying everything and almost nothing works. You know, we are trying super hydrophilic materials, super hydrophobic materials, slippery materials. Proteins still eventually come and stick to the surface. Maybe for a short period of time it's okay, but for longer duration they'll just come stick. So here is an example. Somebody did the study that pre-adsorption of fibronectin that contains RGD sequence increased the adhesion of fibroblast cells. But pre-adsorption of albumin without RGD sequence almost prevented completely adsorption of fibroblast. It was only 5% cell spreading on albumin coated surface and almost 100% of the on fibronectin coated surface. Now, what's tricky about protein surface interactions is that proteins have hydrophobic regions, polar regions, negatively charged, positively charged regions. And no matter what type of surface you are making, protein is ultimately going to come and stick to the surface. We have tried, not we, so many people before us have tried so many things. We have tried things. Protein ultimately gets stuck to the surface. Now, these three scientists have been pioneers in modern protein adsorption studies. So Leo Roman, he's no more, but he has done a lot of work in protein surface interactions, and we will talk. There is a Roman effect also. We will talk about it. That's he figured that out. Dr. Tom Horbett, he's emeritus. He was a professor and now emeritus at University of Washington. Lot of work in 1990s around that time. Another person that I didn't put the picture here is Dr. Buddy Ratner. I don't know if you have heard his name, probably because he has done a lot of work on blood material, protein material interactions, understanding. That big book that I showed you in the start that you can buy, that's the latest version of the book. But the earlier version of the book, Dr. Buddy Ratner was the one who compiled the chapters in that book. Now, other scientists are making it more broader, but he was the original person who started it. Dr. John Brash, he's a professor at McMaster University, and I had the privilege to meet with him maybe 6-7 years ago. We had a big small conference discussing blood material interactions, and I met him there. He has done a lot of work on protein material interactions also. So I'm telling you these names so that if you're interested in the field, if you search their names, you might get some really good basic papers that they were working on in this area. Okay, so when you have protein surface interactions, a lot of things can happen. Okay, proteins can adsorb on the surface, they can desorb from the surface, but I'll tell you one thing, desorption of protein is not very common. Once the protein gets stuck to the surface, it's not very common for the protein to detach from the surface that easily. Because there are so many interactions happening, hydrogen bonding, hydrophobic interactions, that protein gets stuck so tightly to the surface that it's not very easy to physiologically just remove it from the surface. Fractionate mixture of proteins can get stuck to the surface. Proteins can displace some other proteins, not very common, but it's possible. We will talk about it a little later. And actually this phenomenon is known as Roman effect. They can reconfirm, the conformation of the protein can change. They can orient in certain way. They can get activated or they can get deactivated after they get stuck to the surface. Okay. Another thing, protein adsorption is rapid. So as soon as you put implant in the bloodstream, let's say, protein adsorption will happen very fast. Okay. And once it gets stuck on the surface, it's not easy to remove it also. So if you are designing coatings, your coating better starts acting very fast. Otherwise, it will just take over. Okay. I can discuss this later, but let me mention this now. Another issue is sometimes, anti-fouling surfaces are great, but let's say you have a surface and it's preventing protein adsorption, it's preventing clot formation to some extent, it's anti-fouling, but it's not completely anti-fouling. That can be a problem also, because if something got stuck to the surface but it's very loosely bound, because your material is preventing that binding to happen, now that can cause embolism. And what is embolism? So if something gets stuck to the surface, let's say platelets, proteins, there is a little clot, it comes off because it was not that tightly bound, and now it goes in the bloodstream. Now that can be even more dangerous. So making anti-fouling surfaces is great, but if your surface is somewhat working but not properly working and it's not completely anti-fouling, things are still binding, but they got bound and clumped and then got detached, now that's not very desirable. Another thing is protein absorption on surface, it plateaus after certain time, right? It's not that you're going to keep getting protein stuck, right? With time, protein absorption will happen, but then it will plateau. Similarly, even if the concentration of your protein solution is very high, it's still going to plateau after certain time. Okay. Now, most materials absorb same amount of protein or they form monolayer. Okay. Now, depending on the surface, different types of proteins might come and stick to the surface, right? But it's still going to be a monolayer, and approximately that 100 to 200 nanograms per centimeter square protein will get stuck to the surface. And you can imagine now, I hope, even with this much that we are talking, that how challenging all this is, right? How to prevent. Blood has so much protein. The protein is flowing on your surface. You have an anti-fouling surface, but it needs to keep continuously fighting that protein in the bloodstream. This just shows that amount of protein of surface isn't proportional to the concentration of the solution. It's just going to plateau at some amount, and protein continues to absorb until at least a monolayer is formed. You know. Oh, so this work is Dr. Horbett's work that I was showing you in 1996. And see, we also need to always appreciate that is around 30 years ago, and maybe the facilities and understanding of the field was so much less than what it is now. So things done at that time, it's even more, we have to appreciate all the work that was done at that time. I was talking with some students today earlier. Sometimes we feel the work I am doing, it's not so impactful because it's not bringing something to market. But we cannot forget that work we are doing, maybe it's just leading to a publication, but 5 years later somebody can pick up that work and take it to the next level, or 10 years from now actually that can become a product because we helped them understand something. So even if we are designing something today, maybe it's Dr. Horbett or Dr. Roman or Dr. Brash's work or Dr. Buddy Ratner's work that's helping us make the path to come to market with these devices. This is just showing using AFM fibrin adsorption on the surface. Have any of you worked with AFM? So a lot of my PhD was using AFM or atomic force microscopy. It's a pretty cool instrument for multiple reasons. One, so AFM has a small tip and you have a surface and you are scanning the surface. So you can actually with the AFM tip look at nanometer structures on the surface. Okay, so that's cool. But even better is you can do AFM imaging in physiological conditions. See, lot of proteins and all these physiological components, if you dry them, they get denatured. And now if you're imaging them, you're not getting the true picture or true information about it. But with AFM, you can use the buffer and the temperature, physiological conditions, while you are even imaging those structures on the surface. So it's a very, very powerful technique. Another benefit of AFM is, and it actually relates well with the protein, you can measure piconewton level forces with AFM. I have not done AFM in last 12, 13 years, Just last year, we bought an AFM for our lab. I am talking with what I was doing 12, 14 years ago. I'm sure AFM has so much more capability now. So what do you mean by we can measure piconewton level forces? So for example, I don't know if I have A, B, C, D, 4 surfaces, which one binds fibrinogen strongly? I don't know. So maybe we can put this fibrinogen on the tip of the AFM, and then you have these 4 surfaces. You take the tip down, press it on one surface, and then try to pull it. Machine is doing, AFM is doing all this, and you can measure the forces needed to pull it back, detach it. You can do it with all the surfaces, and now you can see for which surface you had to apply the highest force to pull it. So now you know your surface A attaches fibrinogen more strongly than B or C or D. And you can do all this in physiological conditions. So really cool way to understand protein surface interactions. Then I said it a little bit. This study basically showing a lot of people have done it. This is irreversible. You adhere a protein on the surface, then you try to wash it off. And I think Dr. Buddy Ratner, Dr. Horbett, their group did this work also. You cannot wash it off now. It's stuck to the surface. And when in my own way to understand this is, think of like a Velcro maybe, a protein, let's say fibrinogen, it might have these 500, 700 Velcro type interaction points, and now your surface, it got stuck like that. Now you can flow a liquid on it at a very high speed, but there are so many spots on that protein that it's stuck with the surface. Even if you can get detached 2-3 spots, it's still stuck 100 other spots on the surface. So now the protein is stuck on the surface. It's very difficult to just wash it off. Now, another thing that happens is proteins compete with each other to get stuck to the surface. Depending on the surface, which protein will come first or faster, maybe more abundant protein can come first, maybe a protein that's smaller in size can come first. If a surface is more hydrophobic versus hydrophilic, which protein is coming and getting stuck to the surface might change. But like we said earlier, amount of protein getting stuck to the surface is approximately same, in the same range. Okay, Roman effect. So basically the Roman effect is, in our blood, albumin is the highest amount of protein is albumin. Okay, fibrinogen is number 3, I think, or number 4. And I think it's 10 times less than albumin, fibrinogen. Albumin is much smaller protein compared to fibrinogen. And albumin has lesser contact points, maybe 10 times less contact points compared to fibrinogen. Okay, so what Roman effect does is, as soon as a surface is implanted, albumin is rushing to the surface and getting stuck to the surface because it's smaller and it's more abundant. Later on, fibrinogen, which is a much more bulkier protein and it has more spots to attach to the surface, it can come and remove albumin and it takes its place. So this is Roman effect. So highest mobility protein generally arrives first later replaced by less motile proteins that have higher affinity for the surface. And typical example is fibrinogen coming and removing albumin. And this is our problem because fibrinogen is not our friend when we are talking about clot formation or blood getting activated on the surface. Okay, so some facts about proteins. Adsorption is rapid. Within seconds, proteins can come and adhere to the surface. Proteins compete with each other. Plateau level adsorption happens. Mostly monolayer adsorption happens. Mostly the adsorption is irreversible. Yes, Roman effect can happen, protein displacement can sometimes happen, and adsorption can cause denaturation of proteins. Okay. Now we have this really cool paper we can discuss. Let me tell you a little bit about Dr. Robert Latour. He is a good friend and a mentor. He just retired, I think 3 years ago, from Clemson University. He has also done a lot of good work with protein material, blood material interaction type work. And Dr. Shivaraman, he is a friend. When he published the paper, I didn't even know him in 2010. I read the paper, I loved it. 2-3 years later, after this paper was published, I met Dr. Latour and Balakrishnan in conference, and we became friends, and Dr. Latour helped me a lot for last 10 years before he retired. I would like to discuss this paper because you guys will like it a lot. But before that, I wanted to also ask, do you guys know, I hope you understand impact factor of journals, right? Anybody doesn't? Okay, great. Do you guys understand H index? I ask this because, the class I teach, a lot of students don't understand. H index? Okay, can you explain?
So number of times the paper is cited in a particular year, like 1 year from the publication time.
Anybody else?
It's the minimum number of paper a person like has like three. He's an author like in a year he's an author in 3 papers, but the number of papers is being cited and it's calculated like that. Like the 2 is the minimum number of paper.
It's very difficult to explain.
Yeah.
Yes, I think but I'll try to explain it. So, some people like H index, some people don't believe in H index. I don't know. It's not perfect, but it's something, some way to. Now, one thing is number of citations, right? Somebody got cited 2,000 times or 5,000 times or 50,000 times. So that's fine. But the problem there is, okay, what's the problem there? Can somebody tell me? Why can't we just say number of citations? Why do we need H index?
So actually H index takes care of 2 factors, total number of papers published and the total citations. So I think it is the minimum number of citations on the minimum number of papers the author has over the decades or years. So it gives a balanced picture that all these citations are not coming from only one paper which was very relevant. It's coming from a balanced work of the
Great,
author.
yes. So if we just look at total citations, right? Let's say I published one amazing paper that everybody is citing left and right, but all my other papers are not good at all, right? Now I have 5,000 citations, but 4,500 are coming from one paper, and all my other work is giving me 500 citations, right? So that's why H index can be useful. Now what H index is, it's a little bit difficult. Without a board, it's even more difficult, but please listen and hopefully you will understand. So H index is, let's say somebody has published 5 papers, okay? And their one paper is cited 100 times, their second paper is cited 1 time. Maybe you guys can write it so that when I'm telling you, you will understand, because I might forget while saying all this also. Okay, so 5 papers total this person has published. 1 paper published 100 times, second paper published 1 time, third paper published 1 time, fourth and fifth cited 0 times. So 100 citations, 1 citation, 1 citation, 0, 0. What is this person's H index? People who knew a little bit about H index, can you guys answer? So this person's H index is 1. Okay, and I'll tell you why. Now let's say this person's second paper, one is cited 100 times, second one is cited 2 times. Third paper is cited 1 time, fourth is 0, fifth is 0. Now this person's H index is 2.
The minimum number of times.
Yes. Now for this person to become get the H index of 3, their 3 papers, each one should be cited at least 3 times. And to get a H index of 20, you need to have at least 20 papers, each cited at least 20 times. Now if one is cited 1000 times, it doesn't matter. 20 cited 20 times, then only your H index will become 20. Okay, so I mean, I thought you guys should know, you all are in science field, right? So it's not everything, but it's some way to see that, right? Now there is another one that's called I-10 index. Okay, so I think that's also good, because that's saying how many of your papers are cited at least 10 times. How much of your work is cited at least 10 times. It's I-10 index. So I feel the benefit of maybe I-10 index is it's showing little bit future trajectory of the person, maybe, right? Because if you have 10, at least 10 times cited lot of papers, maybe it's showing in coming years they will just keep going up. And it's also little bit showing overall impact of your work, that okay, getting lot of citations is always great, right? But then it's also showing, if you're publishing a lot, are others citing your work? But I have also heard from senior people, sometimes it's not, because what if you are a pioneer in a field? Nobody is citing you because nobody is at the level that you are at. So it's sometimes not true also, this H index, right? Because you are running ahead of everybody else. It might take few years for people to start citing you when they start working on that field. Or your field is so difficult that very few people are working in your field. Now you're not getting a lot of citations. So it's a good number to look at, but you cannot just totally rely on that number. Okay, let's little bit discuss this paper and how should we do it. Let me open this PDF. So is this good? Can you, the paper? Okay, great. Okay, so this paper was published in Biomaterials, which is a really reputed journal in our field. Maybe right now its H index is 12 or 14 or something. Oh, sorry, its impact factor is 12 or 14 or something. So really reputed paper in our field. So what they did in this paper was, maybe read this abstract little bit. So when we talk, I have not read this paper for a while now, but what they did was very cool. Oops. Yes. Okay.
Maybe we could just slide it down.
Okay. I have also given the class notes, whatever we have taught. She's going to share it with all of you. Okay, ready? So I'm going to just summarize what cool stuff they did is, oh, I need to probably,
I guess we can open it on the paper.
will you be able to open it? Like you have that, okay, let's try that. Oh, okay, something worked. Okay, something worked, thank you. Okay. Okay, so what they did is they used different surfaces with hydroxyl group, amine, methyl group, and they put different amounts of fibrinogen on it. 0.1 milligram per ml solution, 1 milligram per ml solution, 10 milligram per ml per solution. And you can see that how the amount of protein adsorbed was so much higher with 10 compared to 0.1, right? Makes sense. Then what they did was, using circular dichroism, you can see how much alpha helix and beta sheets are there in that fibrinogen. Okay. So the first A is 0.1 milligram per ml fibrinogen solution, B is 1 milligram per ml solution, and C is 10 milligram per ml solution. Okay. If you see here, on different surfaces, native fibrinogen, without putting it on the surface, had lot of alpha helix. Okay, Once you put it on the surface, hydroxyl remains almost similar, but other surfaces, amount of beta sheets increase after you put it on the surface. Okay, we are looking at A, which is the least concentration of fibrinogen solution. Same thing happened for B. When you put 1 milligram per ml solution, again alpha helix little bit reduced and beta sheets increased for the same fibrinogen when you put it on the surface. Okay, but when you see number C, the C part, it didn't change. All the alpha helix remained very high and beta sheets remained very low, no matter what surface they tried. Why? Saturation, right? So can you explain a little bit more? You're almost right.
So it's independent of concentration but depends upon the conformation.
Yes. So when they put so much fibrinogen on the surface, there was no place for fibrinogen to open up. So it stayed in its almost native state, right? But then the other interesting thing happened. Now what they did, they put same amount of platelets on all those. So they took the surfaces, they put fibrinogen very low to very high, and then they put same amount of platelets on all those surfaces. And very counterintuitive, the highest fibrinogen surface had least number of platelets. Okay, and the reason is this: because in native fibrinogen state, it's not binding to platelets. The conformation is in such way, alpha helix is high, beta sheet is low, and after we put that high concentration on the surface, we stayed in that ratio, and now the RGD sequence or receptors that were recognizing the platelets, they were hidden and platelets couldn't stick to the surface. So very basic such study, but very well done and very informative, proving the point. You know. Okay. Okay. And we have already talked about this again. I'm mentioning that this is the issue. So protein adsorption, and it's an entropy driving force, right? Proteins liberate structure, water, and that's why they want to just get stuck to the surface. And that's why a non-fouling surface lot of times are very, very hydrophilic, because they bind water so tightly that protein cannot come and just remove that water and takes place, right? The only thing is, for how long? For how long we can just keep fighting that tightly bound water on the surface. Okay, you guys can help me answer this. Don't say the answer, read it. I'll ask somebody. That way you all are reading it. Okay, who will help answer it?
So, with this, I studied it earlier. If the contact angle of the water is more, so the surface is more hydrophobic. So, a more amount of water is limited to a small space. If that is the case, the surface is more hydrophobic. And here the surface B will be more hydrophobic. The surface A will be less hydrophobic.
Okay, and so protein will get stuck more on which surface? A.
So B, because B is more hydrophobic.
B. Because see, the protein is so hydrophobic. In A situation, water is bound so tightly that protein is not able to stick to it. But the hydrophobic surface, due to hydrophobic interactions, it's easy for hydrophobic protein to get stuck because water is not bound so tightly on the surface. But you are right, that is more hydrophobic, that is more hydrophilic. Now, when you have a surface can have hydrophobic regions, polar regions, negatively charged, positively charged. So depending on how the surface is, the protein can adsorb in different orientation, right? Okay, so question for you now. Take 5 minutes, write it down in your notebooks. What are some properties of proteins that affect their interactions with surfaces? What are some properties of proteins that affect their interactions with surfaces? Okay, one more minute, then we'll discuss. Okay, can somebody tell me one property at a time? Structure of protein. Okay. What else?
So you also mentioned the binding site should be open. The nature of the protein. Also.
Amount of protein that is being binding.
Uh, it's like properties of protein, so not just amount.
Orientation.
The region specific charge on
Charge of protein, yes. What else?
Steric hindrance.
Steric hindrance. What?
Isoelectric point without pH or isoelectric point.
That's under charge. So charge, yeah.
contact angle.
Contact angle. It's how will you measure contact angle of, so something else.
Gives free energy.
Hydrophobicity of protein, charge of protein. Yes. There are some things we already talked today. Yes, but there are things we talked today. There are simple things. See,
Hydrogen bonds or disulfide bonds.
okay, so if there are hydrogen bonds or disulfide bonds, how will it impact protein's interaction with the surface?
Make more surface area fit for longer.
Or can we say in easier way, unfolding rate of protein? Because if a protein comes on the surface, it's sticking. The faster it unfolds, faster it grabs onto surface, and now the other protein cannot come and remove it. But it takes more time to open, something else can come and kick it off, right? Okay, there is one more that you guys are missing, and we talked about it today.
Cell binding.
Size of the protein, right? If the protein is very big, it's going to take time for it to come. It might not come, reach, something else has grabbed that spot. So, size of the protein, and bigger proteins have more binding sites also, right? It can come, so a smaller protein can come faster but binds less tightly. A bigger protein can come later but can bind more firmly to the surface. Charge, isoelectric point, like you mentioned, it can absorb more to the surface. Hydrophobicity, more readily absorb on hydrophobic surface. Structural stability, it can fold to a greater extent and form more contact points. And then of course unfolding rate, faster it unfolds, faster it can get firmly stuck to the surface, right? Now, here is another interesting thing. Albumin is almost 4 to 5 times smaller than fibrinogen, 67 kilodalton versus 340 kilodalton. Albumin has 10 times less contact per molecule compared to fibrinogen, right? And all this impacts lot of things like we talked in Roman effect. But albumin is bigger or fibrinogen is bigger? Fibrinogen is bigger. Albumin is more abundant or fibrinogen is more? So that's more abundant, it's smaller, but it does not bind so strongly, so fibrinogen can come and easily replace it. Okay. We little bit talked about it, but this is again showing that unfolded protein lightly expose more sites. Factors that affect rate and extent of unfolding can result in greater surface activity. Less stable proteins are those with less intramolecular cross-linking are likely to unfold more and faster. Okay, protein denaturation on surfaces. Protein structures are not rigid. Proteins are flexible chains that have been coiled, folded and bent to assume particular 3D structure. Changes in microenvironment of protein can alter the conformation of the molecule. So in solution, we talked about alpha helix and beta sheets little bit versus adsorbed proteins can be very different. Alpha helix going down, beta sheets going up. Upon adsorption to a surface, conformation changes, but the protein usually retains some of its biological activity, not all of it, but maybe to some extent, but some is lost. This is just showing that how, if we see the last one, how based on the surface, the difference in protein conformation. You know, for some protein is binding from some way, in the other one, if a surface is more polar or non-polar or negatively charged or positively charged, same surface protein is binding, but the conformation and the way it's binding can change. So Professor Sidlicki is also my very good friend and a collaborator. We have published together. Maybe I'll discuss it next week about some of the work we did together. But this work he did a long time ago, I think. Basically, it's showing, again, it's AFM, atomic force microscopy. Actually, can you tell me what it's trying to show on that side? The right side, mica, APTS, OTS.
So the surface structure of those elements.
Wavelength.
So that thing, that bright thing, that's fibrinogen. Okay, I can tell you that.
The topography of the.
Sure, yes. So AFM tip is imaging the surface, and that's fibrinogen bound to the surface of mica, APTS and OTS. But what else can you tell me from that image?
In which there is the height of the topology is more, there the fibrinogen is not binding more closely to the surface, whereas in the third structure it's diffused, there the fibrinogen is binding the highest among the given surface.
So the third one you are saying it's binding more.
Because the tip of the AFM is catching it to be diffused, not much pointed as with mica or EPTS.
Yes. So mica is a very hydrophilic surface. You know, if you put a drop of water on mica, it will just, so the surface is very hydrophilic. OTS, silane, it's a very hydrophobic surface. So it's basically showing that when they put fibrinogen on this OTS surface, fibrinogen due to hydrophobic interactions, and mica had tightly bound water, it's difficult for fibrinogen to stick to it so nicely, but on OTS this fibrinogen can stick and nicely expand. And if you see, the length of fibrinogen here is longer compared to these two. So it's like more opening up and binding it with more contact points. How many contact points fibrinogen has?
So 770 something.
700 something, right? So more of those contact points are grabbing onto surface now because of surface hydrophobicity,
So it's
hydrophilicity.
also unfolding more in OTS.
It's unfolding more, that's why it's spreading more, right? So now if I put platelets on these surfaces, which one will have more platelet? Don't answer. I'll ask somebody to answer. Okay. So if I put same amount of platelets on all three surfaces, which one will bind more platelets now? Who wants to answer? Raise your hand. Okay, so I'll ask somebody who didn't raise the hand. Okay, what do you think? Which one will have more platelets if I expose same amount of platelets to all three surfaces?
So, if you expose more amount of platelets, the OTS will find it more.
Why?
Because it is more hydrophobic.
So let's say I put lot of fibrinogen now. So forget OTS's hydrophobicity. I covered OTS with fibrinogen. Like now we cannot see OTS anymore. It's covering with the fibrinogen for all of them. Now which one will bind most platelets?
So in that case, if OTS is covering, if fibrinogen is covering all of the surface, so then the binding of platelets on OTS will be less because the original availability would decrease. I think what I understood.
Okay, anybody else want to?
So isn't about the surface, like it's spread it in the third of
Okay, so if I put same amount of platelets on
more
all,
surface to cover, so platelet will bind more in like
to which one?
OTS
OTS. Because
surfaces.
since fibrinogen open up more, conformation more, those RGD sequence are exposed more, now platelets can go and grab onto them. But the other one, since it was hydrophilic, fibrinogen is not liking the surface, it's little bit still closed, and now there is lot of fibrinogen, but they are not exposing their receptors. So platelets, it's not easy for platelets to get stuck to the surface. So OTS will bind most platelets out of the three.
And so also without fibrinogen, like in the blood clotting mechanism, the platelet won't bind. So that way also, in which material the fibrinogen is binding the most, the platelet will bind there also.
So, and if you we can discuss later if you want to discuss after we are done. Okay. Now, next little assignment, you can write the question. Properties of surfaces. Earlier I asked what are the properties of the proteins. Now, what are the properties of surfaces that affect their interactions with the proteins?
So what's APTS particle?
Um,
And also what's OTS?
it's a silent, both are silence.
Kinds of
They both are silence, yeah. So you coated the surface with one type of silane, another type of silane, and maybe they have different contact angles, and I feel OTS contact angle is more than APTS. It's more hydrophobic than APTS.
So in this case, like while they are answering, so in this case we will choose mica hydrogen and less platelet activation?
Okay, good question. So I think when they published this paper, they were not thinking about medical devices or anything. They were just trying to do a more mechanistic study to explain this point. See, the understanding we have now, such strong understanding, it's because of studies like that. So we are not making a medical device of mica. Okay, we are not using mica in making medical devices. Yes, but yes, if you want to make a surface that's more hemocompatible or prevents clotting, we are choosing more hydrophilic surface because this one will chances of clotting is less. But then next week we will talk a little bit about super hydrophobic surfaces. Okay, now if the contact angle of the surface is greater than 150 degrees, it's called super hydrophobic surface. Now the point is the surface is so hydrophobic, like the leaf of a plant or something, that if you drop blood, it just rolls off. So now you are preventing clotting by totally opposite way, that things are not sticking to it because of air pockets and air trapped there, that you are just rolling things off. So it's not that, oh, hydrophobic is bad. Maybe super hydrophobic is good in preventing infection from bacteria or clotting.
So when we say protein interaction, we are aiming the protein should not stick there.
No. So I'm saying properties of surface that affect their interaction. How can I say? I'm not saying protein should or should not stick. So you are saying, oh, I don't want to tell you the answer. If the surface has more of this, it will affect the interaction. It will interact less or more, it's okay, but this will impact the interaction. Okay, one more minute and then you can answer. So see, I love this field because the problem is so simple, but it's not simple at all, right? How to make a non-fouling surface, how to make an antibacterial surface. It sounds simple, but people have tried everything. You can go on Google Scholar, almost every surface has been tested. Okay, who can say the first surface property? Nature?
Crystalline structure.
Smoothness, roughness.
So like hydrophobic surface will attract more protein and the binding will be more firmly and tightly.
Okay, so hydrophobic and smoothness. So topography of a surface, right? So what will smoothness or roughness will do? If you have a smooth surface versus rough surface?
No, the protein will not interact with it more, and the rough surface will interact more.
So rough surface, more protein will get stuck, and smooth surface, less protein will get stuck. And why is that?
Due to the contact that will form between the protein and the
Or the surface area is increasing, right? So that's why you'll bind more. Okay, what else?
It's a lattice shape for the crystalline structure of the metal.
Sure. So I think you guys almost answered. So topography, composition of the surface, chemical makeup of the surface, hydrophobicity and heterogeneity of the surface. See, a surface is not necessarily looking like all same. It can have different domains on the surface, Some domain might interact more or less with proteins. Some domain might want to absorb fibrinogen more. Some domain want to maybe absorb albumin more, right? So different domains can interact differently with the surface also. Okay, surface topography, charge, chemical composition, geometry. Surfaces with more topography feature will expose most surface. Surfaces with grooves or pores have higher surface area compared to a smooth surface. Other surfaces causing roughness. So how you are making a surface, I'll show a slide in five minutes. That changes how the surface looks also. You know, how you're machining the surface. So that can change the topography of your surface. Chemical composition, if there is an oxide layer on the surface, that can sometimes passivate the surface. Ceramic and glasses contains metal and non-metal ions, which can impact how it's interacting with the surface. We saw polymeric surfaces in Dr. Latour's paper, hydroxyl surface versus amine surface. They change how protein adhere to the surface. So hydrophobicity, hydrophilicity, hydrophobic surface typically absorb a monolayer of protein. But one question is, can protein monolayer passivate the surface?
So that is oxidize the surface?
No, not oxidizing, but can you have a surface that absorbs the protein in certain way, in certain conformation, that now things are not sticking to it?
Okay, like evenly adhered.
Yes. You know, protein conformation changes can occur. Super hydrophobic are anti-fouling and repel everything. You know, hydrophilic surface hold water tightly. Proteins can't remove this tightly bound water. Bound water acts as a shield or barrier. Proteins in native conformation. Now protein is not like getting converted to that alpha and beta sheets. It's staying, it's not happily spreading on the surface and sticking. So more beta sheets are coming in terms of that fibrinogen example that we saw. On microscopic scale, biomaterials are inhomogeneous. There can be patches, polymers have microstructures, and all this can impact surface. You know, so many times when we make coatings, films, you look under SCM, surface looks great, you look under SCM and there are certain things, features on the surface. This is just example, don't worry about all this. Basically what we are saying is it's a titanium alloy, but how it was formed, the surface looks very different. This is an example that you have this polyethylene surface, but lamella splitting, or you know, yesterday we talked about how there is a crystalline portion of the polymer and amorphous portion of the polymer. That crystalline portion has these polymer chains stack with each other. If that breaks or splits, it's causing features on the surface. And now that feature can start more proteins to get absorbed, and that gives place for platelets to get activated, and then it can start recruiting more platelets, which we'll talk tomorrow how clotting happens. Here is an example of styrene butadiene block polymer. It has different domains on the surface now, and now each domain is interacting with the surface in a different way. Now, factors that influence protein surface interactions. Protein properties, we talked, size, charge, structural stability, unfolding rate. Surface properties, topography, chemical composition, hydrophilic or hydrophobic regions, heterogeneity of the proteins. Okay, so I want you guys to think about it. When we are talking about protein surface studies, let's say your research is my area and you are designing surfaces, you want to understand protein surface interactions. What are some of the studies that come to your mind you would like to do? Think, just jot down some points, think for 2 minutes, and then we can
So
discuss.
studies as an experimental study.
Experimental studies. I know some of you are computational, but I'm just thinking more bench. On bench, what type of studies would you want to understand your surface? What type of studies would you want to do? Reactivity of protein. Okay, what do you mean by reactivity of protein? Sure, sure. So what would we do to study some of those things?
We will read about the structure of protein, the functional groups, and their reactivity.
Okay. And what will we do bench on the bench? What would we do? How would we do some of those studies?
By structure, oxidation of protein.
I already discussed a lot of this today actually. You guys just have to think what we talked and you can answer a lot of these questions.
For interaction, we should have the idea of protein structure and nature of the surface.
Sure.
For that, we can do empty simulation.
See, I know you guys are simulation. I'm saying what will we do benchtop study?
So extra diffraction, AFM.
Yes. And with AFM, what are we studying? What would you study?
So the surface properties from AFM and maybe the protein properties from the X-ray diffraction or the NMR studies.
And a mass technique.
Okay. Anybody else?
So we can use TEM to study the surface.
Okay. And if we forget the instruments, what would you want to study? Like, I say, okay, my lab is designing this new surface. And I'm saying, oh, my material is anti-fouling or not. What all studies would you want to do on my surface? Forget what instruments you will
So
use.
adhesion studies.
You want to do adhesion. You want to see what protein sticks more and what doesn't stick. Okay. What else?
So there was this diagram graph which AFM, when you talk about if we have a layer of fibrinogen on a surface, so through that like we can go for albumin and we can check for different proteins, like how does it
So
I don't know how to like name it, what
really good. You want to do competition study,
yeah,
right?
comparative.
So you are competing, you are showing, can you put albumin and fibrinogen together on the surface, and then you want to see on A surface what is sticking more versus B surface. What if you can design a surface that sticks more albumin than
You can also
fibrinogen?
take into account the orientation, the angle.
And then if it's sticking, what's the orientation? So yes, so we want to study orientation, we want to study competition. What else? We want to study how sticky different proteins are on different surfaces. Bonds, okay.
So can we go for like antibacterial way?
We will go tomorrow. No, Friday antibacterial. Today let's be anti-fouling.
So, I would like to study like the blood flows drains the entire body. So I would like to go into the physiology and check why doesn't it coagulates anywhere else in the body. Like it flows through the bone marrow, through the tissues, through the ligaments. So there must be a common property lining all such physiological structures and then try to
So you're
replicate
saying
it
why
at some interval.
blood is not clotting at other places?
Yes, sir. Like not adhering at
That's
other
our
places.
Monday's talk.
Yeah.
So that's what my research area is, to study what body is doing
Yes, sir.
and why can't we make surfaces like that so we do the same thing what body is doing.
Exactly.
Yes.
I think this is a straight way, a very laborious way, but the body will only give the answer. Develop such a service.
It has given the answer. We are still trying to make it work,
Yeah.
but we know the answer.
So I would
Okay.
like to study that.
Okay.
What's inside body.
What
So
else?
can we go for a combination of surfaces? Like two materials?
Can you elaborate?
Like in that figure, interfacial or
Oh, you have domains, different domains.
so different proteins will react differently with different orientation and with different degree of binding.
So, thermodynamics.
Can we go for like that?
So, my worry there is, and maybe we can, what I'm just thinking quickly. So let's say you have one domain here, right? One protein is sticking here, one protein is sticking here.
Like when we have taken OTS.
Yes. How is it helping? Think of a medical device, right? You have certain portion of medical device, one domain, certain portion of medical device, one domain. One is sticking more protein, one is sticking less protein, or one is sticking one type of protein, one is sticking one type of protein. How will it help? Let's say you say, yeah, one domain will stick less protein. Then why shouldn't I make my whole device that domain? Why should I have another domain there? And maybe you have some thought there. This is just immediately that comes to my mind, right? Okay, let's come here. So, amount of adsorbed protein I want to understand. Am I making surface that has less surface? Desorption and displacement of protein. Am I making surface that proteins can easily detach? What is the orientation? You guys said protein activity. What if I have designed a surface that after protein sticks to it, the conformation is such that platelets are not sticking to it anymore? That will be cool, right? Thickness of the protein, competition. Can I displace one protein? Can I put albumin more strongly than fibrinogen? Then my problem is also solved, right? Okay. So this is our last slide for today. Just to give you an idea, there are a lot of different techniques used to study protein surface interactions. You know, you can, let's say you have fibrinogen stuck to the surface, you can get an antibody that binds to fibrinogen and it has a fluorescently labeled that you can check. You can have fluorescently labeled fibrinogen that you can buy, and then you can put it on different surfaces and do fluorescent imaging to see what surface is more. You can do QCM or quartz crystal microbalance where you can check the weight. You can do AFM. So there are a lot of different techniques available to just get in-depth understanding of these protein surface interactions. Okay. So I'll stop here, but we can still discuss if you have any questions. But just to summarize everything, for a medical implant, protein is the first thing that comes on the surface. Tomorrow we will talk about blood and platelets, how they come and stick to the surface, but protein is the first thing. Making anti-fouling surfaces is very desirable. Can we prevent protein adsorption from happening? Certain proteins bind more strongly. Roman effect can happen. Fibrinogen is our enemy because it binds to platelets more. Albumin is smaller, more abundant, but it can be replaced by fibrinogen. Certain properties of proteins and surfaces impact their interactions with each other. I think more or less that's what we discussed. Okay. Questions? But we will do antibacterial, we will do learning from the body, all that is coming.
So one thing I can remember that I was reading this paper that was published in Science back in November last year. It was exploring the nature of piezoreceptors on this on our gut, small intestine. It, it said that where there is more mechanistic stimuli, and where in the regions where the piezoreceptors are more frequently stimulated, that those cells have better chances of regeneration and proliferation and everything. So if that is the case with the specific to a receptor, that might be available to the entire all in each and every cell. I might not be sure about this. I might be wrong. So is there something that we can do with our implants, that they give some maybe electrical or certain mechanical stimuli to our specific region that stimulates the surrounding tissue to bind more efficiently to that?
So I know for hip implant type work, you can maybe try these type of things. But let's say you're putting a catheter in somebody. You don't want fouling to happen. So how do we prevent fouling? To increase cell interaction, we can make surfaces porous, like hip implant type work. But the challenge is, how can we put an implant in that does not interact? Like the body, the blood can keep flowing without basically interfering with that tube. That's the challenge. So like she said, blood is flowing in our blood vessels so nicely. We are not getting any clotting, otherwise we'll die. It's flowing through tiny, tiny blood vessels, no clot happening. But as soon as we put any tubing, a catheter, or a stent, or a vascular graft, blood wants to attack it. Proteins want to attack it. They want to stick to the surface. How can we make a surface that stays free of all that?
So but
That's
that's more of a logical response. Like we have a
Yeah.
cut or something.
Yeah.
So it's a human understanding that we are inserting a catheter or something, but the body
Yeah.
is taking it more like a foreign invasion. So it does activates all of the immunity, neutrophil, transmigration, everything. So we need to mimic something which the body considers it's very low.
Yeah.
For that particular. So what if we start making catheters which itself is a biomaterial? Catheter made of a tissue.
Okay.
Which the body recognizes as its own. And maybe we can take, for example,
So one,
a cell of body.
how will you get certain strength in the material? How will you sterilize that material? How can you store that material? Because see, in hospitals, what doctors want is, here is like surgery going on, here is a shelf full of catheters, they'll quickly grab it and then they, even putting a catheter in a freezer and asking a nurse go get a catheter in that time of surgery is, it's too much time and chaotic. They just want very easy process when they are doing it. I mean, this is how we need to think. Yes, you are right, but it's still not so practical solution. But this is the next 2, 3, 4 classes are going to be about. So think about it more. Maybe you guys will give me some good ideas that I can try. So yeah.
So like the umbilical cord. So it stays active even after some time. Like I know people who delay the cord cutting for the genuine transfer of all the immunity and all, even after the birth, given that the baby and the mother are comfortable and everything.
Yeah.
So the material of that umbilical cord is made
Yeah.
and it stays good even in that environment. I know we don't have to insert it back or something and the baby don't have to carry it whole.
But think something like that when you're designing the material.
Yes sir, similar, which is not much prone. But obviously we can't keep it here on the table and then take it for operation. It must be immersed in some fluid for that particular thing, and just the way taking all the precaution we saw yesterday, the hip replacement,
Yeah.
it must be inserted.
Yeah.
So for that example, elastin in our aorta and that cardiac wall,
Yeah.
everything. So it's non-leaky, it's very mechanical. It can take a lot of pressure inside. So what if we make a catheter or something of elastin or maybe a biomimic of it, and those tissues can be easily grown in lab right
Yeah.
now. from the
But
science
see, again, you have to think, can we get the mechanical strength?
Yes.
Can we sterilize it? Can we store it for 2 years? Does it have storage stability of 2 years? Because see, I don't know what's the exact number, but let's say 1 billion catheters are used every year. And nobody's saying, I need catheter tomorrow, let's order catheter, this special catheter to come next week, right? I'm going to buy catheter for next 6 months or whatever time supply and put it on the shelf.
So it would take a lot of time.
And how much will it cost? See, your idea might be really cool, but if the cost of the catheter is 100 times more, okay.
This is like a scaling problem. It's a business problem. but
It's
a science
everything.
problem.
You know, it's a science problem, but it's a business problem.
Yeah.
It's everything. And you are thinking more practically, right? That's how startups, commercialization, you can still publish a cool paper by saying, hey, here is my elastin material, here is my cool catheter. You might get a really good impact factor publication, but that's it. There is no future after
There's
that.
no bench to bedside.
Yes, yes. Okay, guys, thank you so much. And yeah.
Thank you so much, sir, for the lecture. Now we have a break for an hour. We will resume the next session is 2 p.m. at 2 p.m. and now we will proceed for lunch. Thank you, everybody. No, not yet. Good afternoon, everyone. Welcome back to the afternoon session of the Day 3 GIAN course. This session is the first national expert talk of the GIAN course. We have amongst us Professor Daman Saluja as a speaker for this session. Before starting the session, may I request Professor Yasha Hasija, Local Coordinator GIAN and Head of the Department of Biotechnology, to welcome Professor Daman Saluja, Senior Professor, Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. Ma'am, please.
Thank you so much.
Thank you so much, ma'am. Professor Daman Saluja has been working as a senior professor at Medical Biotechnology Laboratory, Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. She got her doctoral degree in 1986 from University of Delhi. Ma'am worked as a research scientist in various capacities from 1987 to 1990 at University of Delhi. She then joined New York University Medical Center, USA, where she worked as assistant research scientist at Department of Biochemistry. Later, she joined Department of Microbiology, NYU Medical Center, and worked as an assistant research scientist from 1993 to 1997. Dr. Saluja joined Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi as reader in 1997 and later became professor in 2007. She was also director of the institute from 2006 to 2010. Professor Saluja has received several awards and scholarship, including National Science Talent Scholarship 1975-1984. She is recipient of prestigious INSA Young Scientist Medal and LSS Kumar Memorial Award in 1989 by Indian National Science Academy. On May 11, 2013, on Technology Day, she was awarded the Biotech Product and Process Development and Commercialization Award of 2013 by the Honorable President of India for her outstanding contribution in the development of novel method of beacon-based PCR diagnostic kit for Neisseria gonorrhea and Chlamydia trachomatis. She was selected for Corona Program Technology Transfer Course 2013 under Indo-US Science and Technology Forum. For the past 20 years, ma'am's lab has been working in the area of medical biotechnology. Her primary contribution is in the development of PCR-mediated diagnostics assays for sexually transmitted diseases. Currently, her lab is working on LAMP-based diagnosis of Mycobacterium tuberculosis. In collaboration with industry, Dr. Saluja has designed a low-cost handheld fluorescent reader. More recently, her group has developed low-cost novel method of qPCR for the detection and quantification of BCR-ABL transcript for chronic myeloid leukemia (CML) and for acute myeloid leukemia (AML, and are also looking into anti-leukemic activity of plant extracts. She is author of 10 patents, applications, and 1 Indian and 1 US patent has been granted. Her laboratory is also working at the regulation of gene expression in eukaryotes using SIN3B as a model system. Professor Daman Saluja has published more than 85 original research papers in international journals. She has produced 20 PhD students, and more than 50 students have done projects under her guidance. She has been invited to more than 40 national and international conferences. May I now invite ma'am to deliver her lecture on innovative tools for advancing point of care diagnosis. Ma'am, please.
Thank you for the nice introduction. And why I chose this, because I thought it was a bedside translational research. So some of the translational work that we are doing in our lab, I will be describing that, and some other work which is going in parallel these days. More recently, in the last 5 to 7 years, we have come out with many more techniques, and as she just mentioned, that handled qPCR method and device was designed in collaboration with other industry. We coming from the um, university and basic sciences, we devised the assay and they devised the machines. So we put it together to for the for easy testing and as a point of care test for the society. So that is our ultimate aim. So what I am going to So I go to, how do I go to slideshow? Yes,
No, ma'am, it's just the issue from here.
I see.
कुछ तो है ना.
नीचे तो नहीं है.
हल्का सा इधर आके बोलेंगे क्योंकि लैपटॉप की पोजीशन आपका
क्योंकि
फेस
ये
नहीं है। अच्छा,
ये
अच्छा,
वायर
इधर से
की
बोल
वजह
सकते
से
हैं।
हाँ मैं इधर से.
Or if you need help, I can help you with the.
He is saying कि
open कर दूँ इसे, so
हाँ.
that even if you move.
मूव दो हाँ ठीक है दो हाँ ठीक है बढ़िया. दिस इज स्लाइड शो में कर दें.
Yes, ma'am. Okay.
Okay. Sorry about that. So, all right. So what we were talking about is why exactly we need this point of care test. And the requirement demand has really increased in the past several years. The reason for that is one, awareness has increased, so people want to get themselves tested. Secondly, even in developing countries and poor countries, and secondly, there are, if you look into the Indian scenario, there are a lot of distances. So you can't always go to the city and get the testing done. You require a lot of expertise for people to carry out PCR, real-time PCR, the assays, even biochemical assays, it is difficult to carry out in remote places or in smaller setups. And therefore, this is very, very important to develop point of care diagnostics. So when we look at what are these point of care diagnostics, they are basically an integration of both, engineering, these are chemistry and microfluidics and microelectronics. And of course, you require back-end software and other hardware to run these devices. So what do these qualify? How do we say this is a good point of care device? WHO says your any diagnostic assay should be robust, which means it should be rapid or quick, accurate, easy to use, portable and low cost, and it should be repeatable assay. So keeping that in mind, a large number of new devices are coming up, and you will actually, if you will see in the last 1 decade, lot of point of care devices have come up. Based on microfluidics and paper based microfluidics, whether it is chip based microfluidics, and they are now we are even connecting these devices with the phone software that you can be anywhere and you will get your results and all that. So of course in developing and poor countries, as I told you, it is very important because hospitals, one, are overcrowded because of high population. Then distance to reach these places, hospitals, is very challenging. Many of the smaller places have poor infrastructure, And often, in poor and developing countries, we have outbreaks. When there will be rainy season, then we will have one set of infections coming up. When it is winter, then another set of infections will come up. So it is always the problem with the society, in the society, to have, it is like a more like a challenge to able to give the right medication at the right time. And because of that, we lose the footfall. A patient, if is told that you come after 3 days to get the test report and then your treatment will start, he or she may not come to the same clinic. By then they might do their own other kind of decoctions and cocktails, or take these medications from over the counter, which has resulted in lot of antimicrobial resistance in the society. These days we are always talking about AMR and multi-drug resistance, and that has come up because of majorly the contributors are developing and poor countries, because the medicines are available on the counter. So this has really become a challenge, and therefore it is very important that we should reduce the time of diagnosis, so that while the patient is still in the clinic or in the hospital, we get the results and then we can start the treatment the same day. Okay. So keeping that in mind, I think the priority by the WHO was put that we must develop good point of care diagnostics, and they have to be approved by WHO or by some society like that in your own country, so that they can be, like in India there is NBL is there, which looks into the quality of any assay that is developed. Even only then it is approved for the use in the hospitals. Okay. So the requirement for any point of care is basically it should be reliable disease quantification, it should improve the follow up, and many of these new point of care diagnostics can even develop, they can give you results on the genetic sequencing in a short time. They are also, many of them are based on proteomics and genomics and transcriptomics data, and nowadays big data is being analyzed to find even the new biomarkers, or to discovery of, for the discovery of the drug, new drug discovery pipelines, so that we reduce repurposing of drug, new drugs, lot of these which is going on in your department also in many labs. Okay. So the advantage of this will be, of course, you can stratify even a given disease into major subtypes. Like for example, I say for example, sexually transmitted diseases, they are caused by more than 20 different pathogens, but some of those pathogens have developed antimicrobial resistance. If my diagnostics test is able to tell me exactly that, okay, this is the patient is infected with X pathogen and it is resistant to this antibiotic but susceptible to this antibiotic or to common other one or two antibiotics, then it is very easy for the clinician to prescribe the drug. And that is what we are trying in Mycobacterium, where we have a huge problem, burden of multi-drug resistance. So now there are devices, I don't know how many of you have heard about Gene Expert, which uses the first line of drug that we use. It has the mutation in that results in the drug resistance, and that is used as a Foster indicator of AMR or MDR in case of tuberculosis. But we need to develop such methods for other diseases also, which are common, and especially in developing countries or in poor countries, because many of these AMR and many of these problems are less in the fully developed or industrialized countries, because they don't have that much problem of infection. But in India and other Africa, even Hong Kong, Bangkok, you will see lot of AMR is coming up, and that is creating lot of problem. So you can really, so even for others, now how many of you have seen glucometer? It is so convenient. A person having diabetes need not go, they can even monitor morning, evening their sugar, and those who are on insulin, they are able to do the test and then decide how many units of insulin they have to take. Okay, the life has become easier, it is better managed, diabetes control management has improved, and there is no inconvenience to the patient. They don't have to keep going to the clinic. So can we really bring such devices which are even useful at home? The patient themselves or somebody at home can run that device and bring, and you have all seen it is so easy. It's just pin prick, and then you put your drop on the strip, and it will give you in within few seconds, it will give you your sugar levels, it will tell you. So similar device has now come for the hemoglobin determination. So it not only does like, especially this is very useful for determining white blood cells, which is a foster indicator of the infection also, and in many cases that is very, very important, especially in leukemic patients. So it can be done at home, nothing, same way you do a pin prick and then you can check. Everything is in the device. So when you put the chip that they have put, it's a more like, there are many versions of this. So even there is a kind of a paper strip kind of a thing, and that can lead to removal of some of the cells, and especially the red blood cells, and then there is a principle by which it determines the white blood cells. So such point of care devices are very much in need for many such diseases. Okay, so there are other point of care devices which are in making, not only determining the hemoglobin, RBC and WBC, but circulating tumor cells. Now from this white blood cells, people are also developing various versions, and some of them have come into the market. Little expensive, like they may be like under 50,000, but there are various versions of that. So depending upon the requirement of the patient, you can buy anywhere from 2,000 to 50,000 device, which is affordable in many cases. But now they want to even look for circulating tumor cell such devices. The patient may not want that at home, but if it is present at any local clinic, it is so easy. If there is a doctor has any suspect for the patient that there is some symptoms and he wants to check whether there are any circulating tumor cells, then this device can measure, and many of these, they basically capture these circulating tumor cells by antigen antibody interaction. So this function as an antigen, you have antibodies, and then there are magnetic nanoparticles which will concentrate it, and then you can measure it. So basically you require a microfiltration where only you will filter out the proteins and other RBCs, and then the principle of hydrodynamics will follow, and then under the various dielectrophoresis will be done, and where you have magnetic beads which are bound with the antigen. So if there are any antibodies you are looking for, you can look for antibodies, or if you are looking for any okay. So I thought there was another figure. So they will, antigen antibody interaction will occur in the magnetic field. So beads will remain stationary, or you can put them in the microfluidics. You can channelize them into the microfluidic strips, and then measure by the spectrophotometric method. Either your antibodies are fluorescence tagged or they are enzyme tagged, and those substrate if you put, then they will give you the estimation. So these devices have become very, very useful, although they have not hit the market in developing countries, but they have been developed, and they will sooner or later they will come into this, and this will become a very good method. And in fact Principle of this technique is what we hear these days, liquid biopsy. How many of you have heard about liquid biopsy? Anybody here? So liquid biopsy is becoming 4 or 5 years back it had become a routine in USA, but now many hospitals in Delhi, at least I know, offer liquid biopsy, and the advantage of that is you are going to take the blood sample. Otherwise biopsy is very invasive. You have to, under ultrasound guidance, you cut the piece, then you isolate, you grow the culture, you look under microscopy, then you do immunohistochemistry to see what kind of antibodies are present, are there tumor antigens are present or not. So it is a long process, and normally 72 to 96 hours they ask for giving the report. Okay, there is one is FNAC, fine needle aspiration, and one is biopsy. So liquid biopsy makes it very convenient because all you have to do is, it is far less invasive because you are taking blood samples. People are more comfortable giving blood samples than getting anything scissored out of there, and sometimes some of the biopsies have to be done in the OT only. You can't do it outside also. So that way this liquid biopsy has become really revolutionized the tumor detection. So some of these tumor cells dislocate and they come into the bloodstream, and you are looking into those cells which have come in the bloodstream, but obviously the number will be less, right? So you have to devise a method to concentrate them to get rid of other kind of molecules, but you should only have those cells which are expressing the antibody, tumor antibody, or outside, or tumor antigen on the surface, depending upon which type of cancer you are looking up. If you are looking for any receptor, then the cell will have antigen on the surface, and you use magnetic beads which will have antibody bound to them. But if there are any kind of tumor antibodies are being produced, the body also has some defense mechanism. Then you look for the antigen, whether this antibody is binding to that. But in liquid biopsy, mainly it is the antigen that is looked, and the magnetic beads and the antibodies are put into that, and then you wash away all the so you do carry out this microfiltration, and hydrodynamic properties will separate some of the cells and from the others, and then you run it on this. This is going to happen on the same device. After this filtration, the magnetic beads carrying those antibodies or antigens, they will move into the microfluidic channels, and then there they will be bound to the to antibodies, and other unbound antibodies will be washed away, and then you do the substrate is lying in the end, and it will give you a particular whatever color or color reaction it has to do. So this has become, yes.
So do you think the detection limit has to be so sometimes, and how often does it miss it? Like, what if there isn't enough? Or
yes, you are very right. So this is like at the for the time being, as I understand, it is being used as a primary detection. Like earlier we used to do FNAC as a primary detection, and then do biopsy, and then histopathology. So and both FNAC and biopsy are highly invasive, and in some cases, like in bone marrow biopsy or GI tract biopsy, it is basically almost like a surgery. You have to give a minor cut to do the biopsy. Oral carcinoma biopsy. So there it is very good, because if you get positive result, you know it is positive. But if you get negative result, then you have to worry about whether you want to call the patient after another 15 days, 1 month again, or you want to do other secondary test and you let him go through the MRI.
Rely on it, but at least
yes, I think it has not come to that robust a condition, but they have already started, because at least you get rid of of major, yeah, where there are more problem in this. For breast carcinoma, they are using it very, they say it is very accurate for breast carcinoma, because it's a very ductal organ. So the antigens, tumor antigens will move very fast to the lymphatic fluid and then to the bloodstream. So it is easy to catch them. And also we need to identify more tumor antigens, because if we have a battery of tumor antigens which we have identified, then we can have multiple antibodies, so chances of missing are much less. But currently we have very handful of antigens like CA123, CA125, and prostate cancer के लिए PSA is there. So we have very limited number of antigens that we are very sure they are good antigens as a marker. But we need, people are now looking a lot into biomarkers, and with big data analysis, transcriptomics and proteomics now, a lot of work is going on in that direction to identify the biomarkers, the genes, proteins that are definitely upregulated in certain types of cancer. And I am sure in another couple of years, we will have this, with the help of AI and machine learning, we will have very good biomarkers which will work across pan cancer, because many of those genes are overlapping in multiple cancers if you do by this transcriptomic data and you use AI facility. So I think we are not far away in identifying more good biomarkers which will be specific. So if we have 3 or 4 biomarkers, 3 or 4 antigens, and they turn out to be positive, we can have them different labels, different fluorescence tags also, and then it will be very easy to identify that yes, there is cancer. First problem is that, but if you look into cancer treatment, medication drugs are not too many. Many drugs are used for different cancers, same drugs are used for different cancers. So I think at least first line of treatment can begin immediately. The early we diagnose and early we start the treatment, the patient has better chances of survival and recovery. Management is much better. So yes, your point is very correct that it is, at present we are still in a dual mode, research come final devices. So this is, I found one of the companies where they were doing many of these things. So they were taking, and I think the engineering students, or if they are here, they will be able to appreciate it much better. So these are the chips that you can see in this and in the figure A, where you have many of these. So I don't know if you can read on the background, but I could not do much about it. So there are cameras, there are microfluidic chip at the bottom. So the camera will move and see capture even the fluorescence or the cells, and whether they have the antigen antibody interaction is occurring, then there will be activity will be there. Okay. So these kind of, it is not only that you are just doing positive, but if using these magnetic beads, if you are capturing these tumors cells which are expressing certain biomarkers on their membrane, then you can capture those cells, elute those cells from those microbeads and use them for RNA extraction, protein extraction, and carry out, these days you can even do with nanograms of RNA, you can do so many PCR reactions and stuff like that. So you can do any kind of downstream processing with those cells. Although this is more at the research level currently, but this is the thought that they are working with, and many laboratories and companies are working now to bring it up to a level where these beads will not only recognize the tumor cells, but then you can also concentrate, these concentrated beads can be utilized for isolation of RNA and the protein, so that we can really come out with the more, and if it is an infection, we can also identify the microbes. In this direction, microbes, actually some more work has been done, and some of the laboratories are actually purifying these microbes from the sample, and by using antibodies against the membrane proteins, and then using that for culture and for the other things. So that is little bit more working in a much better direction. But for cancer, of course, we are still little bit lagging behind and not up to that. So then what do we do after that? Once we have identified these tumor cells, or we have purified these cells, whether they are carrying microbe or anything, then we have to identify things, right? So identification can be done, of course, with proteomics, which requires little more sample, but PCR is one that can be very, very easily done. Of course, you can do PCR directly also. So and because now you have seen during COVID, there have been many point of care devices for the PCR. So PCR is much easier, can be easily used, as well as real time PCR has now become very, very common. So I will first briefly introduce you to people to the PCR. I am sure some of you are very well versed with PCR, but those who are not versed, not familiar with PCR. So this is a nucleic acid amplification technique in simple terms. Okay, so like we use amplifier in music system. This is a specific part of the genome you can amplify to whatever extent you want, it will amplify it. Okay, so how it will do the specific ones is by the specific oligonucleotides that we use. Now it was just little bit of history, like Carimulus, in fact he was on a recreation trip, and that's when he saw the hot spring. It's a very interesting thing, you should see how mind works. So he thought these organisms are also multiplying. That means they are having DNA replication, but their DNA polymerase is very stable because it can handle such high temperatures. And how they must be replicating, it was like out of curiosity, he took a sample of those these thermophilic microorganisms, and isolated DNA polymerase from them, and found the peak, the activity peak, or the maximum activity comes in the range of 70, 65, 70, and like you see a bell-shaped curve normally for any enzymatic reaction. So that's when he discovered PCR. So he took it that if you isolate DNA and denature it at high temperature and allow it to cool and use this enzyme at 65 to 70 degrees, then it will result in the DNA amplification. Now also remember, if you have read in your BSc and MSc, DNA replication always require primer, not RNA transcription. So it DNA cannot start ab initio. It requires a free hydroxyl group at the 3' end, to which the next base will be added. So what he did was, he synthesized the primers for the desired gene and then did this in a test tube. So you will be surprised what you see today, the PCR machines. When he did his first PCR that he standardized, it was 3 water baths. So he would denature at 94 degrees, quickly shift the tube to the 72 degrees, put the enzyme, and then go to allow it to anneal, and then to the reaction. So this he would do for 30 to 40 cycles, and he found huge amount of specific DNA fragment. Okay, so this was very interesting, and that revolutionized the whole molecular biology or biotechnology, whatever you want to say, because now we could amplify the desired genes and we could use them for studying their function, their structure of the proteins, and whatever we wanted to do, kind of mutations that it has in the organism, or we want to modify it, how it happens, what is happening, everything you could study. And that was very interesting, and that led to the applications of PCR, led to the Nobel Prize given to Kerry Muller and Smith. Okay, so this is what I told you, that you have primers complementary to the two strands, forward primer and the reverse primer, and this the synthesis will occur only between the two primer lengths, and it will go. And now, you know, if you look into, all you are adding in this PCR reaction is your DNA, your primers, DNTPs, you just throw DNTPs, buffer, and that's about it. Nothing else, because all your buffer contains the divalent magnesium ion and all that, and DNA polymerase. So earlier it was used DNA polymerase of these, and now we call it Taq polymerase, because from lot of other organisms were identified. They all had thermophilic, from the thermophilic spring, hot spring, so they were also used. And now we have several polymerases that we can use, Vent polymerase and Taq polymerase, and now they are cloned. Their 3' exonuclease activity, 5' exonuclease activity has been deleted, and lot of manipulation has been done with the enzyme, so that you get better processivity and you get better results. Okay. Any questions so far? Any question? Good. Am I going too fast?
No.
Okay. Fine. All right. So this is something, is the basic PCR amplification. So you have one template. From the first cycle you form 2, say you get 2 raised to the power 2, you form 4, then 2 raised to the power 3, you form 16, 32, and by the time you reach to the 35 cycle, theoretically you have 68 billion copies. Although that will not happen, because some or the other agent will start becoming limiting, so that is, but you still get ample amount to do any downstream this. And then once this was worked out, people started looking for more applications of PCR, and many changes were done. This is just the same thing, that you denature at 94 and then you allow it to anneal, the primers are allowed to anneal, and then you add DNTPs and you have the extension. But now we have automatic machines which will result in changing these temperatures. So each cycle has a denaturation step, annealing temperature and extension. They have come up with many more things where you can do your whole reaction at with two temperatures, 94 and 62 or 65, where there will be annealing will also occur at the same temperature, and extension will also occur at the same temperature. And the idea is you make primers such that their TM is also in that range, and also the enzyme has been made in such a way that it can work at 60-62 degrees with full efficiency. Okay. So, but the main trick lies here in the primer designing. So you can develop any diagnostic if you have very specific primers. Okay. So how you decide on the specificity of the primers? One is that the primer should not hybridize to any other site in the genome of the microorganism. Now, when I take a patient sample and I am looking for microbe A, I don't know what all other microbes they have in their sample. Okay. Now the problem comes is because we do not know all the microbes, we only know a handful of microbes whose genomes have been sequenced. Like there are billions of microbes in over there. I'm not saying all of us have billions of microbes in each infection, but even if there are 100 microbes or even 20 microbes, I don't know which 20 are there. So my primers have to be specific. Now when I say my primers have to be very specific, I can only hybridize with the known genome sequences and see my primer does not bind to any of these microbes. So you have to experimentally check that your microbes are specific. How will I do that? If my organism gives a amplicon of about 220 bases, then when I do it with any other microbes for 30 different patient samples, I should, and they are known to carry that infection, I should, from all these patients, I should get only single band of 220 bases. So you initially, when you are developing a diagnostic assay, you develop the specific primers, you standardize the conditions, and then you always look for patients which are already known to carry that infection. This is not a blind assay, this is developing, you are in the developing phase. Once you have proven that your primers are specific, then you start doing blind assays, single blind assays that one person does with a standard gold standard protocol and one person does it with the new method. They don't know the results for the same sample, so it is blind for you. So you are given certain numbers. They know which one is positive and negative by their gold standard, and you have to see whether your results tally with their results or not. Okay, and then you do double blind. Double blind is that you take the samples and they also don't know at present, they also do their methodology, you also do your methodology, and if there is any discrepancy that they are getting positive and you are getting negative, then you use a third test to confirm whether it matches with your test or with their test. This is called discrepant analysis. Okay, and there are certain mathematical derivations which you can use to check the sensitivity and the specificity of the reaction, predictive positive value, predictive negative value. How that predictive positive value is when you say a positive test will always be counted as positive, and predictive negative value is a negative test will always score as negative. So that has to be the best. Okay, if it is 100% PPV and 100% NPV, then your assay is perfect. But nothing is perfect, but WHO takes it that if it is more than 95%, then it passes their test. So any test which comes in the market has to be approved by, like in India we have CDSCO, and in outside in USA it is FDA approval, and then overall if you get WHO approval of your assay, then you can market it in any country. So this is very, very important, and the specificity always come from this. And also interestingly, if I have very short primers, I have more chances of non-specific binding. But I should not have very long primers either, because then also I will have non-specific hybridization. So even if if I am using a 30 base pair primer and 4 have not bound, it does not matter, even then at 60 degree it will remain hybridized, right? So that is a problem. So you have to find, should not be more than 18 to 20 mers, it is good enough. So it should give you a range of this thing, and normally, like we choose 1/4 chance of finding A, G, C, T in any, and 1/16 of this. So we try to look into almost number of G's and C's, and A and T should be approximately the same, so that your TM is around 60. That is what is important. Okay. So, but for some cases we also use universal primers when we don't know any pathogen is present or not, new pathogen, then we use universal primers. Universal primers are ribosomal gene primers, 16S ribosomal for microbes, because ribosomal gene is highly conserved among microbes. And then if I want to look into all the species of a genus, then also I can use certain primers which are specific, some protein which is specific to that genus. I can design primers against that, and then it will give me the how many of them are present. The length, the size of the amplicon may be variable, and that we can use. But then specific is when you are amplifying a single sequence from a milieu which may contains hundreds of other microbes. That is your specific test. All right. So you know forward and reverse sequence, that I have already told you. And Taq polymerase, because it is Taq is from Thermus Aquatics, but there are other which have now come, and this enzyme was the first one to be utilized. So, and component of PCR I have already told you, template and primers, and then the buffer and this thing. So the melting temperature is the most important thing that you have to keep in mind, because melting temperature TM should be around 60, between 60 and 65, not less than that, and beyond that. You can also calculate it by this formula, 4 into G plus C plus 2 into 4 plus T, but these days there are many softwares which will give you the TM, and you must make sure that there are no hairpin loop like structure, and also the forward and reverse primer should not hybridize to each other. They should not form dimers, because they are small molecules, then they will form dimers, they will never give you amplification, you will have poor amplification. Yes.
Is the sequence for which we are making primers against, if just for the sake of the complementarity, it has the hairpin loop structure. How do we synthesize?
So you can always choose nearby area. You can, for diagnostic purpose, that is not a problem, because you can always make, instead of getting an amplicon of 200 bases, you may get an amplicon of 210 or 190, because you can little bit move your primer upstream or downstream on the length. Problem comes when you have to do cloning, because you want full length protein, right? Or you want to clone a subdomain, then there is less problem. But if you want to clone a full length protein, then you have to be very careful. You can't leave N-terminal region for sure, but you can always take little bit extra 3' region, because वहाँ तो termination codon will come, so you will be fine, right? So that is the main problem where you have to take care of that. There are ways to circumvent that. You can put certain, you remember the wobble hypothesis, right? So the third base can be slightly different, so you can make primer like that, but then the primer should not end there. So I can make a primer which is little longer going into the gene, but that base I can change. So it is like the same principle of site-directed mutagenesis that we use. So at the other end, if there is a change in the base, then there will be less of problem, because the last 3 or 4 bases should bind perfectly. Otherwise, you will have non-specific hybridization.
So we are allowed to induce some kind of gaps, not essentially gaps.
But you will not worry about that, because your amino acid will be the same because of the wobble hypothesis, right? When we do site-directed mutagenesis, then we make a change. It should not be the same amino acid, but here you can always do that. So there are like thumb rules, but then variations are always there. All right. So So secondary structure should not be formed, and this is what you have. We are coming back to the same slide. Am I okay? So buffers and thermocycler. This is the most common PCR machine looking like these days. They, instead of having 2 blocks, they have even 3 blocks, by which you can run 3 different PCR reactions independent of each other, independent time pe dal sakte ho, and you can set the annealing temperature and extension and whatever you want. You can program them separately also. Okay, and you can store your program, so whenever you are doing next time, you need not worry about again looking into the programming. Okay, so normally this is what we do, and you normally end up doing 30 to 35 cycles. The only problem with PCR is you only see at the end of the reaction, and it is not quantitative, right? So you will get, you run it on the agarose gel, you see a dark band, and then you say, okay, this is more or this is less, but you don't know how much more or how much. Hey, okay, it is double or maybe little more than double, but it is not truly quantitative. Okay, so it is very approximate, but yes, if you do not get in 30 cycles also any band, then you know it is absent. That particular microorganism is absent. Okay, so lot of changes also came up, as you are as you will realize. If I want to do site directed mutagenesis, or if I want to look into a mutation, especially for AMR, antimicrobial resistance, then I need to design primers which are going to be just looking at that. So in the culture or in the sample, you will have some bacteria which are having a wild type sequence, and there will be some bacteria which will have a mutant. Can you tell them just to be little less louder? थोड़ा धीरे बोलिए, बच्चे डिस्टर्ब हो रहे हैं. So then you have 2 types of templates. So you want to make sure that both the templates get amplified, and you are able to distinguish the wild type from the mutant one. So many new deviations have come up. So you can use this as a you can do nested PCR for that. Hot start PCR is only to improve the this denaturation of the template DNA if it is very GC rich, so that is used. Then there are sometimes you want to do long PCR, so there are separate enzymes which have better prognosis, and then there it comes, the nested PCR is mainly used to look for specific mutations that you want to see in the background of even your wild type. And then of course you want to look into the gene expression, so you have RNA. So once we identified these and sequenced it, we knew where exactly is the splice form. We did Tachman polymerase strategy, and we have quantitatively shown that how in oral carcinoma stage 1, stage 2, stage 3 the splice forms vary. But this is the advantage of doing the PCR, and then you can always couple with real time PCR. It is not always that you have to do, to begin with only you have to do real time PCR. Then another very powerful variation of PCR is restriction fragment length polymorphism. Side you will get the this thing, but many a times your DNA is in very limited condition. So what they do is they amplify first and then they do restriction digestion. Now if there are mutations in the gene, then what will happen is if you do the PCR around that, so you will get and do a digestion with the known restriction enzyme. If you are lucky that your mutation falls on the mutation in the resistant site where it cannot be digested by the restriction enzyme. So, but you can use this in many other devices, like here it has been shown. I think this was for a here they have done after PCR, or they have done Southern blotting, but you can also do it by simple RFLP. So after PCR they did digestion of the whole genome, and then they looked at the probe. So the next experiment can be a probe based, or you can do simply by restriction enzyme. But here, because there were so many bands, they tried to do it by the probe. And RFLP analysis is also used for like sickle cell anemia, and there you have two alleles. One is beta A allele and beta S allele, which is sickle cell anemia allele, and there is a MST2 restriction site is present, and you can see that where the base is changing. CCT instead of G you can have any other nucleotide, and this result in loss of the MST restriction site if there is a change in this GAG sequence, but it has to be, AG has to be there. So if you have in the beta sickle cell anemia, it is CTG instead of GTG instead of GAG or any base. If the G would have mutated, nothing would have happened. But so what happens is, if you take the patient to sickle cell anemia, if you do pedigree analysis, you will see that a person which is carrier of the sickle cell anemia, that means because you always have two alleles are present. So if there is one is normal and one is sickle cell allele, you will get 3 bands. One will not be digested with MST, and the other which is digested with MST, you will get 2 bands because you have amplified that region. Then both the parents have the are carrier, and now the offsprings you will have combination. One will be only AA, which is normal, so you will only get smaller bands. One which is sickle cell, which has mutated, will only you will see the larger band, and again one is who is carrier will have 3 bands. So you can very easily distinguish which child is going to develop sickle cell anemia, right? And even for antimicrobial resistance, you can do, as I told you, for ciprofloxacin resistance. How many of you must have taken fluoroquinones like norfloxacin? And so earlier, 10 years back, ciprofloxacin was the one which was most common drug. But bacteria are also very smart. They mutated, so now ciprofloxacin cannot function in these bacteria. But there is a very simple way to check by PCR followed by RFLP, restriction fragment length polymorphism.
These spots here, like for the mutated BS, here we see only 2 sites and here it corresponds to 3. So are these the MST2 cut sites or these are?
Yeah. So these blue ones are MST cut site. So if I have amplified this region, you can see a blue line. Okay. So you should get this fragment in the, you will get only the large fragment in the sickle cell anemia. It will not get digested with MST. But due to mutation, because GAG site has been converted into GTG site, then you have lost MST. But if you amplify the same from a normal person who is not sickle cell anemic person, then MST will cut in between also. So you will get one fragment will be this and one will be that, right? So you will get 2 fragments. Now if a person is heterozygous, you will get 3 bands because 2 will come from the normal and healthy allele and one will come, a larger band will come from the mutated allele. So you will see 3. So all 3 are carriers, but if you get only one which is larger, then it is completely sickle cell. If you get only these 2 smaller one, that means they are carrier for both alleles, they are normal alleles.
Okay, got it.
Okay. Similarly, you can use it for ciprofloxacin resistance also, and for many other cases we use this as a marker for. But only thing is that there has to be a restriction site in between that region. Then only it is possible, and the mutation must occur overlapping the restriction site. So there are too many ifs. So that is why it is not that common. But otherwise you can use RFLP analysis in this, like for example I had shown you. So you will get from one person to another, the profile will be different, and this is very important in your paternity dispute that we hear. So they do VNTRs, they make the primers for VNTRs and amplify the region in between. So variable number of tandem repeats will help you decide the PCR profile. Multiple bands will come, and if you match the multiple, I thought I had put, let me see, yeah. So you can have it even at the criminal side, you can have it paternity dispute. So one person will have, so if it is, let's say I will explain you for the crime side, the data is there, then you can also extrapolate it for the paternity dispute. So the survivor, you took the swab from the survivor and carried out the PCR using VNTR's primer, so you got a pattern. Now you compare the same set of primers you use for this, and this is the biological sample from the crime site. So crime site primers, there are many bands which are matching with the survivor and then with the criminal. So whichever is there which is matching with this crime scene, that is the person which is. Can any one of you tell me who can be the criminal out of 1, 2, 3? 3, because their bands are matching from the biological sample at the crime scene. Okay, so this is being used. Then many a times what happens is that in the nails, when the person is trying to save himself or herself, then there will be some ruffling between the killer and the person. Then it will happen. In rape case, semen, sometimes they are able to get semen from there, and there they are able to identify the pattern. So this is same thing happens in the paternity dispute. There the child, whatever bands child has, they are either inherited from mother and father. So the one will be their true parents, and the one person there is a dispute, they may or may not be the true parent. So there are 2 sets of parents. So they will do the PCR from all the 4. The child band should match, all the bands that they get should match with either of the 2 parents, like either the mother and the father. But if there are certain bands which are not matching with either of the father or mother, then they know they are not their true father and mother. Okay, so that way paternity disputes can be, this is a very strong evidence which is now even accepted in the court as an evidence. It can be utilized for the search.
One question, the biological evidence at the crime site, we are not sure that it's only of one individual. There could be multiple DNAs present.
Right, you are very right.
So how do we do that?
So in that case, there can be, if there are like that happens.
But mostly there will be multiple because one of the killer or maybe the attacker and one
No,
of the
somewhere at least one or the other suspect they will get, because they take, it is not that they will take sample from only one place. There will be multiple sampling is done. Supposing let's have a scenario that 3-4 people came to kill a person, right? So somewhere or the other they will, all the 3 will leave their mark. Supposing in the ruffling somebody has blood, one person had some bleeding, so uska kahin na kahin blood drop गिर जाएगा. Or even if they are able to catch one person out of 3, कहीं पे उसने अपने आप को बचाने की कोशिश करी, बाइट कर दिया, हेयर फॉल हो गए, बाल खींचे, हाथों के नाखून में आ गया कुछ, तो all these things are taken, and then even if one suspect will be catched, then वो ही बता देगा बाकी के कौन से लोग हैं. But many times they are able to detect more than one from these evidences. Some problem does come in rape cases where there are multiple people are there. So, but there also somehow they are able to decipher the suspects. I think there also they take multiple sampling, and somewhere or the other if they are able to get 1 or 2 people very cleanly, then they are able to catch a rest of the people from that. The person will tell even this was involved, because they want to now escape. They want to either divide their fault or whatever. Okay, but कई लोग जा भी बच भी सकते हैं. So genome mapping के लिए we use RFLP, localization of genes for genetic disorder, risk for the disease, paternity testing, genotyping tests, and forensic sciences for suspects. Okay. Now this is basically, you can also do real-time PCR rather than doing RFLP after PCR. And here also you can do multiplexing PCR. Like this is an example from our work. You can see that with increasing template concentration, you will get lower and lower CT value, which is, all of you are familiar with the CT value after COVID. So that means it will, the graph, there is a baseline and the graph will start rising from there. So higher the concentration, more will be the, so if there are 10 raised to the power 4 templates, then it will come up later than when if it is 10 times more or 100 times more. So CT value will keep on decreasing. Now this becomes quantitative, and this was important in the COVID case. They used to say if your CT value is more than 30, you are almost cured. But if your CT value is in the range of 17 to 25, then you still have very active infection, right? That means you have lot of pathogen in your bloodstream or swab sample, nasal swab or pharyngeal swab, and that large amount of pathogen will give large amount of RNA. आपने तो सेम वॉल्यूम में उसको डिसॉल्व करा, कहीं से भी लिया, तो एज ए रिजल्ट if there are more RNA, that is equivalent to more of viral particles, and therefore you will have a lower CT value. Okay. The devices can be variable, And multiplexing you can do here also, like in BCR able, we are using multiplexing. So we have only BCR, we have only able, and we have BCR able, because we must keep the controls for the patient when we are doing, to be sure that we are looking at the chimeric gene and we are doing and in the background of that. So if you use multiplexing, then your fluorophore should be different. Everybody is familiar with Tachman strategy, right? Good. Now people have, now multiplexing as a limitation, you can maximum you can add 3 to 4 fluorophores. But supposing I say a disease fever, let's say I want to make a fever panel, point of care diagnostic for fever. Now there are so many pathogens which can cause fever, or even for pneumonia, or for sepsis in the hospital, because sepsis is very, very important. Now what do? I need 20 pathogens. So you can use a device which has come up like this. So they have, you put the sample in the middle, and then you have various chambers, right? And each chamber has only two set of primers. So the DNA will go into each chamber equally by the microfluidics, and each chamber has its own primers. So you know, you have labeled the these chambers are labeled from starting 1 to A to whatever B, C, D, E, F. It can have 6 or 8 chambers, and you can at least, if it is 8, you can determine 16 pathogen at a time, because it is multiplexing or duplex or triplex. So you can do it, multiple pathogens you can test in one go, one sample, and it will test for so many of these. This is very, very useful in diseases which have common overlapping symptoms. For example, fever chip, then you know STD, where I have been telling 20 to 20, any amount of, some are viruses, some are bacteria, some are fungus. So you can't have compatible primers for them. So it is better to use different vials with different set of primers. So, and not only this, now they have, or more beautifully, they have come out with devices which are these microfluidic devices on you know, various materials. One is of course microfluidic conventional device. Then they have come out with, I have not seen, but there are paper devices, paper based devices which have chambers like this. How they have partitioned, because you can't see these, but you have many of these are like that. I think I have one figure for that, or maybe not. Okay, so you can have many of these. So there are several pictures on the net you can see for that, and here in this first device that you see on the side, that is a handheld real-time PCR machine which we had been working with one of the company. We have no expertise in electronics, they are doing all the electronics, developing all the, we are only validating the working of that machine with our diagnostic assay, with Tachman strategy, and this machine will be able, people will be able to use it for developing any diagnostic, because it has a chip on which you can put up to 5 microliter of real-time PCR, reaction mix lyophilized, and the assay that we have standardized, it is stable for almost 8 months in that lyophilized form. You seal it under nitrogen and then you can distribute it, so that problem is also not here. In India the temperature shoots up, so you can always transport it at room temperature, or at best at the 4 degrees, which will cut the cost a lot in transportation. Okay. So the idea is that, and the end user has to do nothing. Any technician can isolate the, who can isolate the cDNA, or who can prepare, he can isolate nucleic acids from the sample, and you have to put directly 5 microliter of the sample. Everything which is lying lyophilized will dissolve in that and the reaction will start. because reaction is all programmed on the chip and in the machine. So machine will, it has software and hardware and everything will work accordingly. Okay. So you can, this is just the exponential phase and all that. So these are the single uniplex that we first standardized, and then we carried out the multiplexing, and you can see triplex, green, red and blue. So there were 3 different pathogens that were done, and this was these were the clinical samples, 30 clinical samples, their performance in this assay. So some of them are infected with both pathogen and some of them are infected with only 2, some are infected. Most of them were single infected or double infected, but very few were having all the 3 pathogens in it. Okay, so any questions from here? So I will then talk about the last bit of technique, that is now picking up a large way, although discovered about more than 10 years back, but only now people are becoming serious about its device, and you heard about it in the COVID also, LAMP assay. Okay, so this was first identified only for tuberculosis, but unfortunately amplification is so large that it would result in contaminating the whole lab. You do 10 days LAMP assay in your lab, and then the whole atmosphere is full of lamplicons only for that gene, and you start getting in negative control also, you will start getting the band. So you have to follow very strict conditions that this area is only for putting up the assay, and this area or outside is only for developing the gel or whatever you want to do. So it was not possible. When you are talking about low resource settings, then you can't have dedicated rooms for running the LAMP assay. So it never hit the market. But now people, after this microfluidics coming into and chip assays coming into the diagnostic, people realize that it is very easy to even devise LAMP in a closed container. So if everything is there in the tube and I don't have to run any gels and only the color will change after the reaction, then it is very easy to say it is positive, it is negative. And bandi rehne do tube ko, you don't have to open the tube and just dispose it off. So there will be no spreading. Then there is another thing you can do is use the UDP. So, and you use an enzyme which will degrade all the DNA which has incorporated the UDP. Okay. So this will result in cleaning the whole system from the previous template that is floating around in the lab. So these 2 things had really helped in bringing back the this LAMP assay into the picture, and it has lot of advantages. You really don't need a PCR machine. You have a water bath in the lab, 20 minutes of reaction, you can do it anywhere by even taking a beaker with that temperature. With 60 degrees, you can just maintain 60 degrees for 20 minutes without any problem, and you will get a lamp, this. So tuberculosis, why it became important, because dot centers have to do, for each person who walks into dot centers, they have to do microscopic and then culture assay to be sure that they have tuberculosis. Okay. So LAMP assay can really help. So it is very simple, and why they call it is loop mediated amplification. So with every PCR normally we get straight lines, we get DNA. Here it will loop back and it will keep on looping back, then it will act as a template. So you get lot of loops and like a cauliflower structure. And what happens in cauliflower leaves, one leaf ends and second leaf and starts right before it is ending. The third leaf will start even before the second leaf is ending. Today you go and try to peel the cauliflower and you will see the leaves are overlapping each other.
Cauliflower or cabbage?
Cauliflower.
Okay.
Oh sorry,
Cabbage you meant.
cabbage. Okay, cabbage. So because of that, you will have this overlapping structure. So you end up seeing lot of, and because of this kind of a structure, looping mechanism, you get huge amount of lamplicons. But the only problem with this is the size is variable, whether it is one loop going into another loop, so the size is variable. And if you look at the gel, you see a smear. You don't get bands like in PCR. So one is always doubting whether there is a specificity or not. That is the only problem. Yes, what is your problem?
Ma'am, in the normal PCR, we are getting segments, more generic segments, and here we are getting loop. So what is making it happen this way? What's the
Because I said no, the primers fold back. So you have, but the specificity lies because you have at least 2 sets of primers for amplifying the same gene. In lamp, you can have 3 set of primers. So this figure explains little better. So if you look into F2 against a complementary structure to F2, then when amplification will occur, because it will anyway loop back to the F1. So you can see here F1C, there is so it is little, I will not be able to explain in such short time. So they have primer 1 and primer upstream of that is actually complementary to the other strand, and this is forward, genuine forward. So as a result it will form a loop because it will hybridize right for the next one. So it keeps on getting looped, and that is why it is called loop mediated amplification.
And this is not happening in the normal PCR.
In normal
There
PCR,
we have
only straight
two
primers at the
primers.
2 ends.
Okay.
Okay. And because how many loops are being formed, so you can see in the bottom one A and B, so there can be F and F1C, so there is a 3' end and it will amplify, and here there is a new primer that is binding to F2C and F2, so that also will start. So you end up making lot of amplicons like that, and that is why you get this smear like structure. So people developed as the requirement was, so they didn't want to first run the aerosol because that will result in the forming aerosols of these lamplicons. They are called lamplicons instead of amplicons, and the whole lab gets contaminated. So they developed dyes. They will, if there is a negative sample, then it will be brown in color. If it is positive, then it will green in color. You put cyber green in that. Okay. So, but then people felt that it can be, its sensitivity can be increased and it can be made more user friendly if we spot a very small amount on a paper strip, and it will migrate and it will tell you whether if there is an antibody antigen reaction, then it will give you a control band and it will give you a pathogen band. And you can do, it is called lateral flow dipstick. So all you need to do is put either a drop or you just touch the strip to the lamp assay mix, and you will see it will move, and basically it is again on the strip you have antigen antibody. So test band, you can see here there is an antibody antigen reaction. So when the DNA comes here, it will interact, so you get a positive band, and there is a control band is also there, so you will have this bands are there. So you can see these 2 bands kind of a thing, control band and test band. So control band should always come, test band can come only in the positive sample but not in the negative sample, and here are some results from our lab where you can see those which are positive for tuberculosis, and you can see here intensity. In some cases test line is very dark, that means the person is heavily infected. For other it is light, and for some it is completely missing. So you can see this. So this was a very easy assay, and we could even tell how much we had loaded, one microliter or 2 microliter. And but we found even the tube assay was very reliable, because you could very easily see. So here is the comparison. So upper one is cyber green, different concentration. This was a turbidity assay, different concentration, from 1 picogram to it keeps on going to the 2 nanogram, or I think tube number 1 is 1 nanogram, 100 picograms, then 10 picograms. Tube number 4 is 1 picogram and 100 femtograms, like that. And then the rest, to find out the detection limit. Same is true for this. This is the gel picture where you see all the stream, and here we do the lamp assay, this lateral flow assay, to show that the sensitivity is slightly compromised when we do the test lateral flow assay, unless we put the whole amount. In this we are putting only 1/10 of the amount, because that is much the paper can take over. Okay, so point of care devices for protein analysis have started developing, but we still lack biomarkers, and therefore they will be, once identified, they will be good, faster. And the idea is to again develop lateral flow assay. There was a home test during COVID which was being utilized, and that was partially based on this strategy, where you had IgG and IgM protein detection on using the same lateral flow. And there you don't have to amplify anything, because you are looking for presence of the antigen in the blood and directly putting it on the sample, and then due to capillarity, when it moves, then it has its antibody already attached at the test line. And there were two test line 1 and 2, one for IgG and one for IgM, and you could find out whether the assay you were positive at home. So this was the home test that some people use. But the only problem was IgG and IgM was that the antigen reaction develops only after 4 to 5 days of infection. Okay, so some people in the early time they would miss it, and then the doctor would tell them, but symptoms would not persist, so they will do it after another 2-3 days, and then they will test positive. But the problem is that by then they would have spread it to many other people. Okay, so the crucial aspect to consider in all these paper based assay or strip based assay is the surface characteristics and the capillarity. So in microfluidics we know what kind of fluidic motion we are having, but when we do the paper, there was a cloth based microfluidics device also which came, and many new novel smart materials came up during that time. So depending upon, even for the paper, whether it has come for wood, cotton, flax, jute, bamboo, so each had their separate properties. Because their wetting properties altered and their behavior of these fluids changed. So surface chemistry was very important and porosity was very important. But people are coming out for these, and they are they are putting these for protein markers. They are doing a lysis buffer is put on the protein sample, on the paper or on this device, and then the proteins through porosity and due to capillary action they move towards the site, and you can spot the antibodies for them and try to identify what all samples are what all antibodies are available and are present there. So, so that has given rise to 2D and 3D microfluidic strips where you can do photolithography. I'm sure many of you are familiar with that, and you can do plasma etching, wax printing, inkjet etching, and you can have many of these devices. So since we have some cancer C-reactive protein, so people are trying using that as a model system to test whether they are able to identify C-reactive protein using these similar strategy as we had seen in the microfluidics in the beginning. And you have gold nanoparticles, and you can hook them to these nanoparticles and try to develop these devices. Okay. So many more futuristic research, however, is required, because you need to handle, one thing is the small amount of the fluid should be used, because many a times it is not possible. I will give you an example. If you go to a leukemic patient and you want to take the blood, 2 ml of blood, for doing your genuine real-time PCR, so you need to isolate RNA and cDNA. It's very difficult from leukemic patients to take another 2 ml of the blood. Normally you will hardly get blood out of them. It is not that they have zero blood, but it is only whitish fluid. There's very low hemoglobin in there, and there is very little plasma in that, and it is really pathetic in some cases to take blood from children. There you can't even get 2 ml. You can hardly get 500 microliter from children, because some of the cancers are childhood leukemias, so there is huge problem. And even for other tests, you might have noticed that in pediatric section, many a times they take the blood from the foot of the child, because you cannot pierce here, the veins are too small. You can't take out the blood, and you will always hear them screaming to the top of their voice. Even for vaccination, they are scared of injections, and then taking out blood is very difficult. So the idea is we should have even less invasive, like if we can use saliva, if we can use nasal swabs, if we can use urine, then it will be very good. But then if we have to take blood, then it should be pin pricks or very small amount of blood, so that the patient is not harmed, or patient can give that much of blood. So minimum handling, small handling of the volumes, and that can only come with your microfluidics. It is very important, whether it is on paper or on the device. So we should go to femtomolar detection sensitivity from millimolar. We should have multiple marker panels. It should not be single panel, because the single protein or single PCR site or gene, because that can at times give us wrong results. Okay. And then of course low cost, long lasting material. That means we should not be carrying them in -20, -80 stuff. And there should be ease and self-containment should be there, and robustness and accuracy, and so this is the ideal situation. So we have to work towards reaching these ideal situation, and the future lies with you people. You know where to go and what to contribute to the society in large. And thank you for your attention. I'm glad all of you came on the holiday to come for that. All right. So any questions? So no questions?
Ma'am, is there any device, microfluidic device for, as a non-invasive obviously, for extracellular vesicles, EVs?
I don't know like one specific ones, but lot of microfluidic devices are coming up now in market, and depending upon their intake, how much volume they can take, people are trying, but they are still exuberantly costly. So they have not come to, daily, like real-time PCR machine that I have told you. It is portable, but they are not ready to sell it less than 5 lakhs right now, because they have put lot of money in their R&D. So unless they become equally competitive and less expensive, exactly, more demand, but that will happen gradually. And to the microfluidic device or the chip, it does not matter where from the sample is coming.
No, no, but for the convenience, like EVs are very high in urine and saliva sample, so it's easier.
Yes,
No
yes.
blood prick and nothing.
Yeah. So on the microfluidic device, you are going to put one preliminary extraction you will do. Whatever is there, like even if I take any body fluid, I probably will do a strong spin, collect the EBs and suspend it in the lysis buffer, because the DNA has to reach. So I have to lyse. Even if the protein is there, I have to put protein lysis buffer. So somewhere I have to lyse it, because macro structures of will not be able to penetrate the microfluid fluids of the, or the porosity will be, if it is that porous, then it will not be able to fractionate the material. Yeah. But who knows, they might come out with better devices in future, because on chip lysis they are trying to do. Gene Expert for MTB, sputum is very difficult to handle, but Gene Expert has devised a method by which you put directly the sputum first in the chip, the sputum will be lysed, the DNA will be extracted. You don't have to do anything. The machine will keep running for 30 minutes. They have the lysis buffer inbuilt and washing buffer, everything, and they have a kind of a gel sink in which all the fluid will go into that and get stored there, because in the end you are going to throw anyway that chip, and then only the DNA will move to the PCR chamber. So they have device, but it is their trade secret, it is not out. Nobody knows how to do it. So, right. Any other question?
Ma'am, in microfluidic devices, do we need to prepare our samples in advance, or they can be easily?
No, this I just told you in Gene Expert MTB, and now they are coming out for other infectious also diseases. They are coming out with that same device. I know they have couple of more in their catalog. But I don't know, like I showed you this paper based kind of or textile based devices. I don't know how much they can do it like that. Although in their picture they are showing that you put the protein extract. So, but you have to isolate the extract and do the lysis, because then only the protein interactions will separate, or you put the cells and do lysis. So things have started moving in this direction, but they have not, everything has not come to the market yet. Many of them are actually still in R&D phase, but in advance. And the advantage of devices is, they don't take as much time as FDA drug approval, because they are in vitro. So they only have to come and show that their device is as sensitive or more sensitive than the existing one, and it is easier to use than the existing device. So that if they are able to demonstrate through their device, then it can be launched in the market. And if it is good, the marketing will take over. Right. So that way, those which are in R&D, we can hope that in the next 5 years, we will have many such devices for diagnostics. Like liquid biopsy didn't come very, many years back. It is quite recent, liquid biopsy. And it is already being done in India. These private hospitals are using liquid biopsy. So it is picking up fast. So I'm sure these devices will very soon get launched. Some of them I am told that they are in the this validation, clinical validation phase in USA and some in Europe. They are already in the validation phase, so they might hit the market very soon.
As for now, I have just seen one microfluidic device that was using Rajiv Gandhi for CTC actually, one microfluidic device. Other than very few market
Yeah, and then MTB, you can say Gene Expert. Gene Expert kits are there now because once they have developed that device, now they are just adding more and more diagnostics to that. So, but we need many more because they can't capture the whole market anyway. Because इतना तो किसी भी कंपनी का मैन्युफैक्चरिंग रेट नहीं होता. We need lot of such devices. Any other? All right. If not, so fine.
Thank you so much, ma'am, for your time. And as our Honorable Vice Chancellor often says, that we have the national experts here who are the best in their fields, who have been delivering their decades of experience in just 1 or 2 hours. So we must thank ma'am for her time and sharing her experience.
Thank you for inviting and letting me interact with these people. It's always interesting if the students are interacting.
Thank you so much, ma'am, for this thorough presentation and this very interactive section. Now, may I request Professor Yasha teacher to felicitate Professor.
अरे अभी तो हो गया फिर.
No, this is felicitation. Just take it. I'll do it, ma'am. I'll do it. I just take care of the rest. Thank you so much for your time, ma'am.
Okay, thank you so much.
Thank you.
Thank you. Wonderful.